Yes, I admit all. So welcome everyone. Welcome to this first appointment of the year. And first of all, from all the crew of the European Webinar Series, we really want to wish you a great and hopefully peaceful 2026. It's our pleasure today to have Peng with us. Let me just share exactly do you see my slides? Yes. So just to remind you that this is an initiative created by the three teams dislocated all over Europe. We are in Switzerland with the team Claire Compagnon, Giorgio Corsi, and Gloria Colombo. I don't see, sorry. I lost something weird with my Zoom is happening. Sorry, but do you hear me?
Yes.
I had the disconnection, sorry.
Yes, I can hear you.
Great. Sorry, I just lost the connection completely. So I was just saying with Gloria Colombo, Felipe Espinosa, and Giorgio Corsi. And we have the team led by Amanda Sierra in Spain, with Nora Bengoa, Fede Soria, and Isaac Buendia. And the team that recently joined us with Morgan Thion from France, with Marta Pereira and Sara Viguier. So as you can see already, we have a list of seminars from the next semesters. And if you don't have the chance to attend one of those, you can also follow them offline. There is this link in the repository where you can have easy access. And today we have the pleasure to host Peng from Fudan University. Federico Soria will give the introduction and Marta Pereira will curate the Q&A. You can write your question in the chat as usual, or you can also ask them directly if you have access to your microphone. So Fede, I stop sharing. The floor is yours.
Thank you, Rosa. Peng is a distinguished professor at Fudan University in Shanghai. His research is centered on the turnover and replacement of microglia in the central nervous system. He has pioneered microglia replacement for the treatment of neurological disorders. In 2020, his team achieved the world's first efficient microglia replacement in animal models. And in 2025, his group published the world's first clinical trial of microglia replacement therapy. His key research findings have been published in high-impact journals like Science, Nature Neuroscience, and Neuron. And Professor Peng has received numerous awards, including the Asian Young Scientist Fellowship, among others. Okay, Professor Peng, the floor is yours, or the room is yours in this case.
Yeah, thank you. Thank you for the very kind introduction. And also, I feel very honored to be invited here to this really wonderful webinar. My name is Peng from Fudan University in Shanghai, China. Today, I'd like to talk about a little bit different because we are all talk about the function of microglia and the role of microglia in the homeostasis. Now I'd like to talk about a little bit different. I'm going to talk about microglia replacement. This is not a meeting, this is not a topic only focused on the animal study from the bench side. It is a topic about from the bench side to the clinical therapy. So the first question, what are microglia? As all we are big fans of microglia, we should know that microglia are the important immune cells which are originated from the yolk sac. During the very early developmental stage, they migrated into the brain rudiment. After the formation of the blood-brain barrier, these cell populations can independently develop and maintain their homeostasis. In the adulthood, there are no microglia progenitor cell at all. And there is a very important receptor essential for microglia survival. Its name is CS1 receptor. When researchers or when us utilize Pell's compound, which is a specific inhibitor for the CS1 receptor, we can see that during a very short period, the majority of microglia could be killed at a very short period. And people reason that if we kill the majority of microglia first and we remove the CSF1 inhibition, what would happen? People believe that for a long time, the microglia cell number should be maintained to a very few number because in the adulthood there are no microglia progenitor cell. However, when people remove the CSF inhibition, they surprisingly found that the microglia can rapidly repopulate and regenerate and repopulate the whole brain. The microglia, even within a very short period, the microglia repopulation is first reported from the Zach Mattis lab at DZNE in Germany. They first reported the phenomena and they believe that the uniform microglia repopulation are actually derived from the monocytes in the blood cell. Therefore, in 2012, they have the first origin hypothesis we called monocyte origin hypothesis. And 2 years later, another group from UC11, they don't believe so. They believe that the uniform microglia are originated from the differentiation of nesting positive progenitor cell in the brain. Therefore, 2 years later, another hypothesis have came out. Because there are two totally different hypotheses of microglia origin, so we reasoned that what is the origin of the repopulating microglia? To this end, we first utilized nesting Korea AF14 as a genetic model for mapping the cell phase of nesting progenitor cell. However, we found that actually there are no uniform microglia originated from the differentiation of nesting positive progenitor cell. Therefore, we excluded the nesting cell origin hypothesis. And also, we utilize the parabiosis for fast mapping the peripheral blood cell and found that even in this process, there are no repopulating microglia are originated from the differentiation of peripheral blood cell. Therefore, we also excluded the monocyte origin hypothesis. And also, we found that the other cell type cannot give rise to repopulating microglia in the brain. And finally, we demonstrated that all the repopulated microglia are solely derived from the residual microglia in the brain. During this process, they can transiently express nesting. However, the cell phase has now been changed. Okay, after we know how microglia regenerate, so the next question is, so what? When we need to pay a lot of such efforts to understand how microglia regenerate, or in another word, why repopulation is very important to study. I will take this as an example as a very important immune cell. As very important immune cells, microglia are involved in different processes in the body, including the development, the homeostasis maintenance, and the disease. If microglia carry some gene mutations, the pathogenic gene mutation will lead to the CNS dysfunction and disease. So we reason that if we can replace mutated pathogenic microglia with anogenic gene normal healthy cells, so we can treat the neurological disorders. The idea is simple. However, the key point is how we can achieve the efficient microglia replacement. For the traditional way, people utilize the traditional bone marrow transplantation for the microglia replacements. As we can see that if we introduce the GLP positive bone marrow cell into a GLP negative mouse, we can find the GLP positive microglia in the GLP negative receiving mouse brain. However, all the GLP positive cells are only localized in the very specific brain region, and at the whole brain, the cell replacement efficiency is only 2%. It is a very low level. Obviously, it is far from the satisfactory of the cell therapy-based requirement for replacement. Therefore, we need to find another approach. If traditional bone marrow transplantation is not a good idea, how about we directly transplant microglia into the mouse brain? So we did that, and we found that for the traditional microglia replacement, the cell replacement ratio is much lower than that of the traditional bone BMT. Therefore, both of the traditional BMT and traditional bone microglia transplantation are not a good idea for the cell therapy. So to this end, we need to find another approach to achieve this. So how can we achieve this? We reason that if we want to achieve an efficient and high-level microglia replacement, we need to allow the allogenic transplanted cell to rapidly regenerate and repopulate the brain of the host animal. Okay, so therefore we get to the key points: regeneration and repopulation. We compare two different conditions. For the first one, in the normal condition or in the physiological condition, the turnover rate or proliferation rate of microglia is relatively very low. However, during the microglia repopulation process, the microglia regeneration rate could be 300 higher than in the normal condition. Therefore, why between these two different conditions, the microglia regeneration rates could be such different? To understand this question, we need to look at our brain, or the mouse brain. In the normal condition, microglia are fully tiled the whole brain. Microglia has a very high capacity for proliferation. However, the proliferation capacity could be inhibited by their neighboring microglia. We call it network inhibition. When the neighboring microglia die, with the removal of the network inhibition, the microglia could able to proliferate. Therefore, during the proliferation repopulation stage, with their neighboring cells are all gone, the network inhibition has been totally removed. In that way, the surviving microglia could be rapidly proliferate, migrate the whole brain, and repopulate the whole brain. Therefore, we reason that if we want to achieve a efficient high-level microglia replacement, we need to create a microglia-free niche. To test our hypothesis, we utilize the bone marrow transplantation for the nutrition, but the difference is we utilize the CSF1 inhibition to kill the majority of microglia in the recipient mouse brain and create a microglia-free niche. And we found that with a microglia-free niche, the majority of microglia could be replaced by the GLP positive microglia. And the microglia replacement ratio is higher than 92%. This is for the first time researchers or our human being can achieve a efficient high-level microglia replacement in the animal model. We call this approach as a microglia replacement by bone marrow transplantation, or MRBMT for short. In our lab, we call it MIS-BMT. Therefore, with a microglia-free niche, we developed a new method called MIS-BMT can achieve a very high-level microglia replacement as we show here. So far, we have a tool or strategies to achieve this to achieve a microglia replacement. The next question is how we can utilize MIS-BMT for the neurological disorder treatment. Therefore, we need to go back to the CS1 receptor. As I have been mentioned, this receptor is very important for microglia survival. Therefore, the function is very essential for this receptor. If CS1 receptor or if the CS1 gene have some pathogenic mutation, it would lead to a very severe disease. We call it ALSP. ALSP is a fatal disease. the patient would suffer the motor dysfunction, the cognitive decline, and they have a very high level for the seizure and the Parkinson-like phenotype. It is a fatal disease. The average survival time is around 3 years to 6.8 years. Before our study, there are no clinical cure for this disease. But because the pathogenic mutation is specifically expressed in microglia, so we reasoned if we can utilize MIS-BMT to replace the CS1 mutated microglia with the allogenic CS1 normal healthy microglia, we can treat the disease. To this end, we have to do the preclinical study first. We first compared all the gene mutation in the CS1 gene, and we can look at the two different hotspot mutation expressed in two different tyrosine kinase domain. The name of the hotspot, the gene mutation is E633K and I794T. And we based on the human hotspot mutation, we generated two animal strains. The names are E631K and I792T, respectively. I will take this as an example. We can introduce the GLP positive and CS1 normal the bone marrow cells into the CS1 mutated microglia or the LP animal model as we show here. And we can see that including the cortex, hippocampus, and corpus callosum, all the brain regions, the majority of microglia could be replaced by GLP positive allogenic microglia. The replacement efficiency is more than 19%, which means we can utilize MIS-BMT to achieve a very high level microglia replacement, which means by this way we can have a very high level gene correction in the whole brain. Therefore, the next question is whether the microglia replacement-based gene correction could lead to disease treatment benefits. Therefore, we compare the major pathological features of ASP. First, as CS1 mutation, the microglia cell number has been reduced. After the microglia replacement by MIS-BMT, the microglia cell number have rescued to have received to the normal condition. Secondly, in this disease, they have a very severe many pathology. We have compared 4 different parameters of many pathology, and we found that after gene correction by microglia replacements, the many pathology has been well rescued, and also the axon swelling have been reversed. Therefore, in our study, we demonstrated that the microglia replacement can have a very good therapeutic effects in ASP and preserve the brain structure. The next question is whether the brain structure preservation could lead to a improvement to the animal brain function. Therefore, we perform the behavior test, and we can show that because CS1 inhibition, the motor activity of ASP mice is very bad. However, in contrast, after the gene correction by microglia replacement, the motor function of AFT mice have rescued to the normal condition, and also by microglia replacement, the cognitive function have very well preserved. Okay, after that, we can revisit all the approach of microglia replacement. As I have mentioned, three different approaches. Only Mr. BMT can achieve a very high level microglia replacement. However, for the traditional BMT, the microglia replacement ratio is very low, very few. So we revisit the procedure of Miss BMT. There are two key points. The first one, we utilize CSF inhibition to create a microglia niche. By this way, we can remove the lateral inhibition from in the endogenous mouse brain. In addition, we utilize the bone marrow transplantation to introduce the donor cells. If you look at this procedure very carefully, and we can easily tell that the mixed BMT is actually a CSF inhibition plus traditional BMT. For the ALSP, the gene mutation is harbored in CSF1 gene, which means this disease is very special. This disease is a naturally CSF1 inhibition state because the gene is already mutated. Therefore, which means in this disease, we don't need to apply the pharmacological inhibition. It is a naturally CSF1 inhibition state. Therefore, we reason that because the unique mutation in this disease, whether the traditional BMT in this disease is equivalent to our mixed BMT procedure. To test our hypothesis, we utilize traditional BMT to introduce GFP-positive bone marrow cells into the ALSP mice. And we can see that all the brain regions that we have observed, the majority of microglia are replaced by by GFP-positive allogeneic microglia. The replacement ratio is around 18%. It is a little bit lower than the death of Mr. BMT. However, it is still a very high level. And also, by this approach, we can have a very good therapeutic effect in the ASP. Therefore, we can have two conclusions. The first one, because the CSF inhibition in this disease, the traditional BMT is equivalent to mixed BMT, or in another words, because the disease is very special. In this disease, the traditional BMT is a simplified version for mixed BMT. And the other things, by the simplified mixed BMT, it can also have a very good therapeutic effect. Therefore, these results suggested that it would be has a chance to in the clinical trial we utilize this approach for the clinical trials. Okay, all the above mentioned approach or the study are conducted in the animal model. So to us, the most important thing is whether the therapeutic effects of the mixed BMT or the microglia replacement can indeed utilize in the clinical trials, in the clinical therapy. To this end, we conducted the clinical trials. First, we can utilize the MRI to access the brain structure and the brain pathology. And also, we can qualify the brain pathology based on the MRI. We use the approach we are using is called Sandoz Stability Score, or S for short. As we can see that there are 4 health control. Each solid dot means it is a male, and the empty dot indicates it is a female. And we can see that because there are no brain pathology, the Sandoz score, the S scores are 0 for the health control. And for the ASP patients, and we can see that there are very severe brain structure impairments in the brain. also for the T1 structure and the T2 flare and the DTI structure phases. And we can see that the triple S is very high. And if we look at the development of the triple S change, and we can see in the 6 months and the 12 months follow-ups, the disease progression is very rapid. So we asked, what would happen if we apply the microglia replacement? Therefore, we replace the pathogenic microglia with the CS1 normal cells in 8 patients. Each 8 patients, including the female and the male and the female, and no matter the severe score at the very beginning is as this or at a very severe stages. All these 8 patients have a very stable triple S. The triple S level is no longer developed. And if you look at the triple S trend, as we can see, there is a significant reverse trend. Therefore, we demonstrated that the microglia replacement in the clinical trials can completely halt the disease progression in the brain. Then after that, the next question is whether the brain structure preservation have the benefits in the brain function. To this end, we utilize 4 clinical evaluations to evaluate the motor activity and cognitive function. And we can see that if we didn't do the microglia replacement, the motor activity and the cognitive function declines very rapidly. And after the microglia replacement, each of our 8 patients has very good performance in both of the motor activity and cognitive function. Both of these do not decline. They have a very stable level for the 6 months, 12 months, and 24 months follow-ups. These are all data, so we look at another angle, the video. This is a very typical patient. At the beginning, these patients can freely walk, but due to the very severe disease development, only after 12 months, this patient totally lost her motor activity. He cannot independently walk. He can only sit in the sofa and behave like that. As we can see that it is a very severe and desperate disease. So we can look at another patient. At the beginning, this patient can walk. However, he cannot wave his right hand because of the motor impairment. And we conducted the microglia replacement to correct the gene mutation. After 24 months, the motor activity has been very well preserved, and also the motor activity is much better than that before the microglia replacement treatment. Therefore, we for the first time achieved the microglia replacement in the in the animal model, and also we for the first time utilized microglia replacement in the clinical trials and get a very good therapeutic treatment. It is not only the first in human, it is also the first in class. And also, this is the only clinical cure for the ASP. After we achieved that, we have revisited the literatures in the field. And one paper published in 10 years before have our annotation. Because in this paper, they reported a very special patient. In 2020 or for the 26 years before, this patient has been misdiagnosed as with MLD. It is another disease. And at that time, in 20,000, sorry, it is not 2020. It is 20,000. It is 2000. In 26 years before, this patient has been misdiagnosed as MMLD, and at that time, this patient has been treated by traditional BMT, which is a standard treatment procedure for MLD. And a few years later, the clinicians found that these patients actually are not MLD. These patients are actually a patient with ASP. But, but the clinicians very surprisingly found that this, this patient has been misdiagnosed as and has been mistreated. But after the treatment, the disease progression has been stable for more than one decade. For more than decade. Therefore, the clinicians don't know what would be happen, and they propose that it would be possible that they should be introduce some CSF1 normal gene in the peripheral system. It can treat the disease. Based on our study, we found that the explanation is totally wrong. These patients, it is ASP because the very special, the very unique, because the disease is very special. In this disease, the traditional BMT, it is actually a simplified version for microglia replacement. Therefore, why the disease has been halted for more than 10 years, it is because these patients is very lucky. Because after the misdiagnosis and the mistreatment, these patients has experienced a right treatment for the simplified mis-BMT. With the gene correction in microglia, the disease progression could be halted. And also because of the disease, we can have a very good faith that the microglia replacements have a very long therapeutic effect for diseases. And also, our study also explained that some of the small population clinical cohort by the bone marrow transplantation have the good treatment for ASP patients. Okay, now we call this approach for the disease treatment and the enhancement as a microglia intervention strategy for therapy and enhancement by replacement. The name is very long, but we don't need to remember the full name. We only remember the short name. We can call it MR for short. So what is MR? MR includes our MR-BMT. MR-BMT is very good, but MR-BMT is not our only MRs. Why? Because the disease is very sophisticated, and for the clinical treatment, we have experienced different clinical conditions and clinical scenarios. With different clinical scenarios, we need to utilize different treatment strategies or approaches. Our MRs, including MR-BMT, MR-PB, and MR-MT, each of the MRs have their own unique advantages and can fit for different clinical scenarios and requirements for disease treatment. After our publication, we have released our very detailed protocol in STAR protocols to help the community to moving forward. Okay, after that, we can revisit the history of microglia replacement. Before 2020, researchers cannot achieve a efficient microglia replacement. We could call that time as pre-replacement era, as shown here. In 1988, researchers found that the bone marrow cell has the potential to differentiate into microglia. Therefore, we can utilize the bone marrow cells as a donor cell for replacement. Only have the replacement donor cell is not sufficient because if we want to achieve a replacement, we need to remove the endogenous pathogenic cell first. In 2011, researchers of the pioneer study demonstrated that the CSF1 gene, the microglia specific CSF1 gene, is essential for the survival of microglia. So 2 years later, researchers can utilize CSF1 inhibitor BLZ compound to pharmacologically deplete the microglia. And that way, we can remove the endogenous pathogenic microglia. Only have the cell removal is also not sufficient because we need to achieve a replacement by the bone marrow by the allogenic donor cells. Based on our study, we rely on the phenomena of repopulation. In 2012, Zach Martin's lab at CCNE, they first observed the microglia population and they proposed the monocyte origin hypothesis. And 2 years later, another group proposed a nest cell origin hypothesis. But in our study, we found that all of the cell origin hypotheses are not correct. In 2018, we demonstrated that all the microglia population are solely derived from the local proliferation through the self-renewal. And only after that, we can only understand the mechanism of microglia population, we can achieve a efficient microglia replacement in the central nervous system in 2020. Therefore, at that time, we have opened up the microglia replacement era. And in last year, we reported the therapeutic effects of the microglia replacement in clinical therapy and found that demonstrated that microglia replacements have a very good therapeutic effect in the neurological disorders. Therefore, our lab, the contribution of our lab has shown here. We have we have the we have our very unique contribution and the pioneer study for the microglia replacement. And the most important thing is that the microglia is not a specific method to treat a specific brain disease. It is a totally and novel cell therapy. It is a total new idea for the new cell therapy. By microglia replacement, we can treat different CNS disorders. ASP is only a start. So what else diseases we can treat by this approach? So we can take AD as an example. If we take GWAS analysis, we can have some of the genetic risk factors for sporadic AD. The top genetic risk factors, including ApoE4, adenine, and TREM2, all of them are relevant to microglia, especially for TREM2. TREM2 in the brain is specifically in microglia. In 2018, a study from Hua Qixu's lab demonstrated that the TREM2 is a receptor for A beta. If TREM2 has mutation, it will lead to the impairment of microglia to the A beta recognition, migration, and engulfment. In that way, it would lead to accelerate microglia the progression of AD. Therefore, in our first paper, in the discussion part, we reasoned that if we can utilize microglia replacement to correct the gene mutation in microglia, we can have a chance to treat or slow down the progression of sporadic AD. So the question is whether it works. The answer is yes. 3 years later, a follow-up study from the Stanford University, they utilized our MIS-BMT to help us demonstrate our idea can have a very good therapeutic treatment in the AD mouse model. In addition to AD, recent years, a lot of studies from the community have demonstrated that our microglia replacements have very good therapeutic effects in different disease models in animals. Therefore, it is means that the microglia replacement idea we proposed and first achieved so far has become a very important hot topic, including for both of the neuroscience and the cell therapy communities. Okay, so in conclusion, we first studying how microglia repopulate. In the very beginning, we demonstrated that all the repopulated microglia are solely derived from the proliferation of the remaining microglia in this process. And after we know the mechanism of microglia repopulation, we can for the first time achieve the microglia replacement in animal model in 2020. And a few years later, we applied the microglia replacement in the clinical therapy and have the very good therapeutic effects as shown here. This is for the first time. This is for the first in human and first in class microglia replacement therapy. And also, we last month we have published a very long perspective review. In that review, we revisited the history and the idea, the concepts, the mechanism and the treatment approach of microglia replacement. If you are interested in microglia replacement, please approach this paper. You can find almost everything about microglia replacement. This is published in Cells Cell. Okay, with that, I'd like to thank all the heroes in my lab. Without them, nothing would be happen. There would be no such a microglia replacement cell therapy strategy can treat the disease for the human patients. Also, I'd like to thank my long-term collaborator, Professor Yinxia Rao at Fudan University. And the clinical trials was collaborated with Professor Li Cao. It is a very brilliant clinician at the Shanghai People's Sixth Hospital at Shanghai, affiliated to Shanghai Jiao Tong University. And the electrophysiological studies was collaborated with Professor You Sheng Shu at Fudan University. And also, as the very last but not least, I'd like to show our gratitude to all animals sacrificed in this study. Thank you so much, and like to take any questions. Thank you.
Thank you so much, Bo. It was an excellent talk. Yes, we have maybe a few people start writing the question. I can start asking you. I have a couple of questions because these are very impressive data for the impact that the replacement approach that you are using, both on the mouse model, especially in the human patients. So I wanted to ask you if you have now the experience with your mouse model with the mutants, if you have a sense of how much the time matters. So because you have done the replacement at 2 months of age of the mouse model. If you would have acted much earlier, would have had a different impact, or is the time after the replacement that matters the most? What is your feeling?
Yes, thank you, Rosa. I just think this is a very important study. I will get some data just for a couple seconds. In our study, the most of our study, we conducted the microglia replacement at 2 months old. It is quite a relatively early stage for the disease, and we can have a very good therapeutic effect as we can show here. And also, we have ever tried the replacement the time point at a little bit later at around 8 or 9 months old. At that time, at that time the animal has a very severe has relatively severe disease symptom for this disease, or each much late or much later or even much later. I think it is at around 12 months old. I think if we start the disease treatment at 12 months old, it also have a very good therapeutic effects for disease, but it is not as good as uh, at a very early stage. But the therapeutic effect is quite good.
I see, because
The
I
key
noticed.
point is the key point is we can apply the disease treatment at the relatively late stage. However, if we conduct the disease treatment earlier, we can have a better the disease treatment.
Yeah, I was asking because 2 months is already past the developmental window. And
Mhm.
I wanted to have a feeling if acting very early would also have different kind of effect. Because I realized that some of the tests, if I'm not wrong, the Y maze, for example, you could not rescue the phenotype. So there are certain phenotypes that still are not rescued by the replacements, as opposed to many others, like the motor behavior that you show. Um, so is it because of different developmental windows, perhaps, that are not, that have long-lasting effect?
Yes, I can see that we have applied different uh we have applied different behavior tests. I think eh, is this Oh yes, this is this is cognitive performance by
Exactly, exactly.
by different approaches. I think this is the first one is NOR, the second one is YMAS, and we can see a very good therapeutic treatment improvements in the
The results
in the
developed.
NOR, but we only show a enhancement trend for
Mhm.
the for the YMAs, but it didn't reach the statistical significance level. Yes, this is for the animal model. I think we need to report it our results very honestly. So we didn't hide anything, even though
Absolutely.
there is a non-significant result. We also put it here.
No, those are beautiful results anyway. I was just wondering whether there is something that is cannot be rescued if it's not in the intervention is not in the critical window. So that was my question in that direction. But of course, you have rescued many phenotypes that are very important anyway. So
Yes, I think the most important part is for the clinical therapy because in the well, it's because the most important part is for the clinical therapy, and we can see as shown here, both by MOCA and MSSE, the cognitive decline has been perfectly halted as shown here.
no, that's beautiful. And then just link to this, do you ever follow up of these individuals, these patients, after these
Mm,
2 years of?
Yes, we are doing that, and the both of the brain function in the cognitive function and motor function has very well preserved.
Mhm.
Compared to the non-treatment control, they could be very well preserved.
Amazing, yeah. Thanks.
Thank you.
Marta, you want to read the question in the chat?
I don't.
There are no questions yet, I guess.
I see one. But probably
I
was
do.
only sent to me for some reason.
Oh, I don't have it, sorry.
Yeah, it's true. So I was
Okay.
but I can read it loud. So excellent talk. I might miss this, but if the repopulation is achieved by the remaining microglia and not monocyte derived, then how does transplant work? Presumably, it's the healthy monocytes from the bone marrow transplant that is repopulating the brain after endogenous microglia are depleted. Thanks.
Oh, yes, I think this is a very good question. In the normal condition, in the physiological condition, the researcher from the fibrosis lab demonstrated that the microglia, I think it is here, and also the microPRIS lab, they found that in normal condition, the microglia, the renewal of microglia is not originated from the circulating blood cells. And we demonstrated that even in the repopulation, it is not from the blood cell. However, in our approach, we are quite different because we are not because in their paper, in this study, the the so-called progenitor cells are peripheral blood. However, in our approach, we utilize the bone marrow cell. Bone marrow cell is different from the circulating blood cell. So this is the major point. And also, we concluded how the strategy of microglia replacement as shown here. The principle is that if we want to achieve a microglia replacement, we need to achieve a microglia-free niche to remove the natural inhibition as shown here. But if we want to introduce the transplanted cell, we need to stop the CS1 inhibition. At that time, the remaining cell can rapidly repopulate in around 5 to 7 days. It is a very short period. In this very short period time window, both of the bone marrow cells or the peripheral blood cells have no chance to infiltrate into the brain and replace the endogenous microglia. In that way, microglia can very rapidly repopulate, and the circulating blood cell can have no chance to go into and replace the free niche. Therefore, we need to create a prolonged microglia free niche. We need to provide a lot of space and a lot of time to allow the endogenous cell to infiltrate and to repopulate. Therefore, in our study, we utilize the myeloablative conditioning, including the laser radiation or the chemotherapy. By these approaches, they can inhibit the proliferation of the surviving microglia, but and without affecting the donor cell. In that way, they can maintain a prolonged microglia free niche. In that way, they have provided a longer enough time to allow the donor cell to go into the brain and to proliferate and occupy and replace the brain. Therefore, we need to treatment like that to achieve the microglia replacement. And also, we also concluded that the other approaches of the microglia replacement, all of the underlying mechanism and key points is to create a prolonged microglia free niche as shown here. And now it is only a mixed-based strategy can already utilized in clinical therapy. And theoretically, we doubt that all the other approach can only utilize in animal model and have a very little chance to be applied in the real clinical trial. Thank you for your question.
Maybe I have one for you. So thank you very much for your talk. It's very impressive data set regarding really what we can achieve in human and regarding these devastating disorders. I had a question, maybe not related to the timing, but also in mice, when you are using the PLX depletion, you always have a few resistant microglia cells. Do you have an idea of what makes them resistant and how maybe you can trigger those guys to avoid maybe to do irradiation and so on to kind of have your microglia-free niche?
Okay, thank you very much, Morgan. I think it is a very good question. In the early 2020, a study published in Life, they utilized selenium B to harvest the surviving microglia by fax and utilize the single cell ion sequencing to look at the transcriptome of the surviving cells. In that study, they found that these surviving microglia have the monocyte-like phenotype features. Therefore, in that study, believe they claim that the surviving microglia has a kind of the progenitor-like phenotype with a monocyte-like phenomena. However, myself have looked at the single cell ion synthesis data, and we found that all the cell population they claimed do not express any microglia specific gene, including AF1, CSCL1, P2Y12. They don't express. Even for the selenium B, they use that as a marker to pull them out. That cell population is CD11B negative, which means they get that conclusion because these cell population are actually the cell contamination. They are not actually the bona fide microglia, surviving microglia. It is from the contaminated cell. And in our theory, we need to based on what's that the microglia niche, because when create a microglia niche, for example, like that, the surviving cells are the neighboring cell disappear, the highly proliferative capacity can be invoked. In that way, they can rapidly proliferate. However, when we deplete microglia, in the same time, it is also creating microglia free niche as shown here. When we kill microglia simultaneously undergo cell death and cell regeneration. The surviving cells are actually, it is a balance of the cell death and the cell repopulation. Therefore, and cell death and cell regeneration. Therefore, what we show, the surviving microglia are actually a balance in the whole brain. The surviving microglia per se are not the internal property of the surviving microglia per se are not unique, but it is a balance of cell death and cell regeneration. Thank you, Morgan, for your question.
And maybe, Bo, just a last question on my side. What are the potential risks of this approach? Can you tell us what you think would be the side effect of replacements?
Okay, this is a very good question. In this approach, we need to meet the two criteria. The first one is the donor cell should be the CS1 normal gene, because we need to do the gene correction. And the second thing is for the HLA compatibility. We need to utilize the HLA fully matched donor cell to avoid the immunorejection. Even with the HLA matched donor cell, it could have also a chance to have an acute ghost virus host disease or GVST or GVHT. This is one of the risk. The risk is not arise from the microglia replacement per se. It is arise for all of the endogenous transplantation, including the organ transplantation and the cell transplantation. But the acute GVHT GVHD could be well controlled in the clinical therapy. We have the treatment for controls, and also for the longer microglia, for the longer risk factors, especially for the clinical trials, we need to took a longer time to observe. So far, we didn't observe very severe long-term the side effects for this disease. Thank you for your questions.
Thank you.
There's a question in the chat. It says, what are your thoughts on using iPSCs-derived microglia as a donor population?
Yes, I think so. I think we can, in the future, we can utilize iPS cells as a donor cell. In that way, we can extend the availability of the donor cells. The answer is yes, but we need to do it one by one because before our clinical therapy, there are no examples for that treatment. We cannot put a lot of the condition in the first clinical trial. We need to one by one. After the successful treatment for the first case, we can in the future we can utilize something like gene correction, IPS, and something like that. Thank you for your questions. And also in this review, we propose that we can utilize the embryonic stem cell or the iPS derived microglia as the donor cell, as shown here.
Great,
But
thank
maybe
you.
yeah, sorry, just to add maybe kind of a comment on microglia maturation and the fact that if you make a replacement really during adult stages, and then if you use do you have kind of a comment or a remark to just discuss a bit how you can have a short-term maturation of microglia and adaptation to their local niches once you graft them in the brain? Have you checked a bit maturation profiles and how they can you get my
Yes,
point? Yeah.
yes, I think it is also a very good question. Actually, because the autology of the replaced microglia are different from the York-cell-derived microglia in normal condition, so the characteristics of the replaced cell are not identical to naive microglia, as we can show here. This is naive microglia, and for our mixed BMT cell, they are not the same. We have three different approaches. Mixed BMT and mixed PB cells are not identical to the naive microglia. However, for the mixed empty replaced cell, it is identical to naive microglia. Therefore, because of the different autology of the replaced cell, the function and the characteristics could be a little bit different from naive microglia. And we can compare that. For the microglia specific gene could be much lower than the naive microglia. This is not negative. This is just downregulated. However, for the macrophage specific genes are kind of the upregulated in this mixed BMT and mixed PB cells. Thank you for questions.
Thank you.
Thank you so much, both. Thanks to everybody for attending. And I wish you a nice evening or a nice day, wherever you are. And I'll see you next month. Bye.
Yeah.
See you.
See you. Bye.
Bye-bye.
Bye.
Thank you both. Bye-bye.
Thank you. Thank you very much.